lecture, chapter 15 on the digestive system and nutrition. I'm only going to do a little bit on nutrition, probably not very much, just because this is not a nutrition course, and some of the content that this textbook uses to teach nutrition, I don't really agree with. It still talks about BMI, which is not really a useful tool for determining health, and other aspects of diet culture that I just don't want to teach or feel comfortable teaching. So we're not going to be really getting too much into nutrition on the lecture, but we are going to get through the digestive system. So just to start off with, the different aspects of the digestive system obviously is going to be going through digestion, both mechanical and chemical digestion, to allow the breakdown of foods and absorbing of nutrients. That's also the function of the digestive system. Mechanical digestion, that's things like chewing or breaking things down into smaller pieces. Chemical digestion uses enzymes and things to help break down into smaller molecules that can be absorbed. The body, you can't just eat, I usually go with like a chicken breast. You can't just eat a chicken breast and your body absorbs that completely. It has to be broken down to smaller and smaller microscopic components before you can absorb those nutrients. The digestive system itself is this collection of tubes and organs. If we think about just the tubes, we call that the alimentary canal, or the digestive tract, the GI tract. It's all the same thing. So if you hear people talk about the alimentary canal, they're talking about that tube from mouth to anus. That is one continuous tube. And then you have the accessory organs, things like your liver, your pancreas, salivary glands are going to be producing secretions to help with that process. And so when you include the alimentary canal plus the accessory organs, then you get the digestive system as a whole. Here is just a view of that. You can see again that tube from mouth to anus. Technically, anything inside of this tube is actually outside of your body. The GI tract is continuous with the mouth, which is a mucous membrane, your esophagus, stomach, your intestines. Those are all mucous membranes. And if you remember, mucous membranes are exposed to the outside. And so you might think like, well, it's in my small intestines. It's here in my abdomen. That's not outside of me. But technically, because your mouth and your anus are exposed to the outside environment and there's no real solid barrier between those tubes, yes, you are just kind of this one long tube from those, and that is still outside of the body. It's a evolutionary trait to help make sure that something that we ingest stays outside of us. It has to be absorbed before it can actually be considered inside of us. And so if you ingest something that's harmful for you, it has to pass through those layers. It's a safety guard. As I said, the alimentary canal is a muscular tube going from the thoracic and abdominal cavities. It's a passageway from mouth to anus, and it's about 8 meters long. So it's a long tube. I have a friend who's a mathematician, and so he likes to say that mathematically humans are donuts because we just have the one hole. That's a weird thing, I know. Hopefully you're laughing with me and not just freaked out by that. All right, so the wall of the alimentary canal, when we think about the tube, that muscular tube, it actually has 4 layers to it. So again, this way of making sure that any pathogens that we ingest aren't just immediately absorbed into our body. They have these layers they have to go through. That's not the only reason for these layers, but it is one of the good benefits for it. So the innermost layer is the mucosa. And again, I said your oral cavity is a mucous membrane. Your GI tract is a mucous membrane as well. So this is truly secreting mucus to help keep things nice and lubricated and moving along. This mucosa is what's in contact with food. It's what's in contact with the digestive material. It has a layer of epithelial cells, right? So those are those kind of covering cells. Underlying connective tissue, keeping things anchored, and then smooth muscle, a little bit of smooth muscle, and we'll see what the smooth muscle does in a little bit. It helps to protect tissues of the canal. Since it has epithelial tissue, it's able to do secretion and absorption of dietary nutrients and enzymes. And sometimes this mucosa gets modified to help increase surface area. So we've seen on the outside of cells things like cilia and microvilli. We'll see some of those here as well as other features. The second layer is the submucosa, so just below the mucosa, submucosa. It has loose connective tissue, blood vessels, lymphatic vessels, nerves, and glands. This is the area of absorption. So nutrients have to pass through the mucosal layer, get into the submucosal layer, and this is where they can come in contact with blood vessels, transporting nutrients away. Deep to that is the muscularis layer. So it has two layers of smooth muscle. This is independent from the smooth muscle in the mucosa layer. That really is just to help with secretions. The muscularis layer is what actually pushes food through the canal. It has an inner circular and an outer longitudinal, so kind of going with the length of the tube. And so these two muscles can work in conjunction with each other. The circular muscle acts as a sphincter muscle and compresses or constricts, whereas the longitudinal pushes. And so those two actions together can kind of give this a movement of material through the GI tract. The outermost layer is the serosa. There are some parts of the GI tract that don't actually have a serosa layer, but the majority of it does. This is acting as this outer layer, and it's a serous layer, so rather than being a mucus layer, which is on the outside of the body, technically, or exposed to the outside, a serous layer is an internal membrane, kind of like in your lungs. So this outer serous layer, we call it the visceral peritoneum. So visceral on the organ, peritoneum is what refers to the abdominal cavity. It helps mostly to anchor things in place, protecting underlying tissues, secreting serous fluid to reduce friction, and like I said, just kind of helping to organize things where they're supposed to be, attaching it and anchoring it to different structures in its area. Here you can see some of these layers. So again, starting with the outermost, serosa is this thin serous membrane. You have the muscularis layer with some nerves in there. There's the submucosa with its blood vessels. This green thing is a lacteal from the lymphatic system. And then you get into the mucous membrane, the mucosa layer. And you can see that it has these villi that are in here. And we'll see that villi are these projections that help to increase the surface area for increased nutrient absorption. And then each of these villi, it's still a continuation of these mucosal cells, of these epithelial cells. And so if we kind of take this section here and we can look at it, you can see this is the top of the villi. So the whole thing is the villi. And then each of these cells right here are these individual epithelial cells. And if we zoom in on one of those epithelial cells, you can see that they have microvilli on top of them. So when you look at this kind of large villus, you have to also remember that on every one of these cells covering and lining the villus, they have microvilli. So it's just millions and trillions of little projections to help make sure that more nutrients can be absorbed, optimal nutrients can be absorbed. So let's talk about movement in the tube. Movement is part of the physiology of digestive tract. If it did not move, then we would not have a functioning GI tract. So two different types of movements. There's mixing and propelling movements. The mixing movements are called segmentation. So this is when the muscularis layer will kind of push food back and forth, and we'll use that circular layer to isolate it off. So it literally starts to just, if you think about food, we'll call food different things as it moves through the GI tract. So apologies if I sometimes get that a little mixed up or don't quite stay congruent, and sometimes I just call it food. Technically, food is what you chew, what you swallow, what goes into your stomach. But once it gets into your stomach and it starts to mix with other stomach juices, it then becomes chyme. So after digested material, that's the other general word we use, after digested material moves through the stomach, we call it chyme. And so essentially it kind of gets turned into this kind of very mixed mass. Kind of think about how they make chicken tenders, chicken nuggets, right? They kind of have that pink slime kind of thing. That's what I always think about. I hope you're not eating during this lecture. It will not be pleasant. So basically what happens is that you have, once you're digesting material, you have just kind of this just mixture of stuff. And so your body goes through segmentation to help make smaller and smaller pockets of that mixed material to be able to break it down even easier. Because we'll see that the more you can break things down, the more surface area gets exposed, and the easier it is to go through the complete digestion absorption process. Segmentation, that's a mixing movement, mixing it with chemicals, enzymes, things like that. The propelling movements, we call it peristalsis. So peristalsis is this contraction of the circularis muscle, the circular muscle in the muscularis layer, that helps to move things down. So it kind of constricts. And if you kind of think about like squeezing toothpaste out of a tube, how you kind of push it along, you're kind of, you use your fingers to kind of squeeze the tube nice and tight at the bottom, and then you slide it along. That's kind of like peristalsis. It's this rhythmic movement pushing masses along. These aren't the only two movements. We'll see. We can also talk about the MMCs, the migrating motility complexes. This is when, let's say it's in between meals, and all of a sudden you just hear your stomach and your intestines give this like, and it's this really big gurgle. That's when it's just like, oh, we've been sitting here for a while. Let's just move a bunch of this stuff along. So you just have this really quick movement of material across through your digestive tract. Here you can see some of these examples. This one is that segmentation. Like I said, it's kind of mixing back and forth, breaking things down, all this digestive material. And then this is peristalsis, where you're having this constriction, this rhythmic kind of passing down constriction of the circularis muscle, propelling this material. So now basically we start, that was kind of the general functions of the digestive tract. We're going to get into each part of the GI tract now. So we're going to start in the mouth, go all the way down through the anus, and along the way we'll talk about accessory organs and what type of digestion is happening in these areas. So the mouth is the first portion of the alimentary canal. It functions in mechanical digestion through mastication. Mastication is chewing, and it also does have an effect in chemical digestion through the release of salivary enzymes. We call it the oral cavity. That's the chamber between the palate and the tongue, the palate being the roof of the mouth. That's your palatine bone. We can also name the space between the lips. It's called the vestibule. The tongue is a thick muscular organ. It is skeletal muscle. It's voluntary muscle. It has these projections called papillae to provide friction for moving food in the mouth. If you kind of are looking at your tongue, if you ever look at the tongue, you might notice that as you go towards the back, it gets a little almost scalier looking, or like you can see that it has kind of these bumps on it. Those are the papillae. And then the taste buds sit alongside of the papillae. So this is all for helping to move food. What's really cool too about your palatine bone, so if you use your tongue and you kind of rub the roof of your mouth, you'll feel those ridges, those palatine ridges. When you're chewing, what you're actually doing, and you probably don't realize it, is that as you chew, at a certain point, you're going to use your tongue and you're going to push the food that you're chewing up along the roof of your mouth and use those ridges and your tongue to help break food down even more, to go through more mechanical digestion. So the next time you chew something, notice that like at a certain point you kind of push the food up there to help really break it down, to make it easier to swallow. Unless of course you're just swallowing food whole, in which case you're bold. So the taste buds are along the sides of the papillae. You do not have certain regions that detect certain tastes, by the way. It's not like sours at the tip and sweets on the back. Taste buds are interspersed all across those papillae. They are more located towards the back of the tongue, but it's not like certain areas only are geared to detect certain tastes. That's not how it quite functions. Lingual tonsils, these are part of the three sets of tonsils in the body. Lymphatic tissue at the root of the tongue to help filter any ingested material. There's just a nice little picture of that. So the lingual tonsils would be under the tongue here, but these in the back, those are your palatine tonsils. So when you go to the doctor and he makes you say, and they make you say, it's the palatine tonsils that you're showing off. Soft palate with the uvula hanging down below, and then hard palate more anteriorly. As I said, the palate forms the roof of the oral cavity. It has the bony hard plate and the softer and the posterior soft palate. The projection of the soft palate is the uvula. That's the thing that's hanging in the back of your throat. It helps to close the nasal cavity during swallowing. So it's one of those processes. The epiglottis covers the trachea, and the uvula covers the nasal cavity. The palatine tonsils, these are the ones in the posterior nasal cavity, or kind of in between the nasal cavity, oral cavity. I'm sorry, I said that wrong. The palatine are in the back of the oral cavity. The pharyngeal are the ones that are in between the kind of oral nasal cavity. They also, you see this name adenoids. So when the pharyngeal tonsils, if they continuously become inflamed and infected over and over and over again, that's when they actually call them adenoids, and they typically get removed. So if you hear someone say, oh, I had adenoids, or I have adenoids, that's what they mean, is that their pharyngeal tonsils are routinely infected, and they, again, might have those removed. There's a nice depiction of that. Again, you can see these lingual tonsils down here, palatine tonsils, and then the pharyngeal tonsils right up here. kind of in that posterior lower nasal cavity. All right, let's look at our teeth. So your teeth have different parts to them. There is a crown that projects above the gum. So the thing that you brush with your teeth, that is called a crown. That's why when you go to the dentist, if you crack your tooth, you get a crown molding, or not crown molding, that's in your house, but you get a crown on your tooth. And then the root is the part that's below, and that is what's connected to the jawbone. So your tooth is a bone. The structure of your jawbone and your tooth, your maxilla in your tooth, or your mandible in your tooth, that is a joint. It's called a gomphosis. It's a very supposed to not be a movable joint. It's an arthrotic joint, but the root is the part below the gum line. The crown of the tooth is covered by enamel. That's the calcium salts. This is the hardest substance in the body, harder than normal bone would be. Normal bone has a bit of a give to it. Your teeth do not, and it's covered by this enameling. Under the enamel, we have the dentin, and then deep to that, you have the central pulp, the cavity. That's where the blood vessels and nerves are. A layer of cementum connects the tooth to its periodontal ligament that then connects it to the jawbone. So again, this gomphosis joint has all these different structures to it. If you think like a synovial joint has the membrane and the fluid, a gomphosal joint has these other structures. These are just the number of teeth, where they are, and their general function. So it's a good thing to kind of learn, especially if you're going into kind of dental hygiene. Make sure you this should be bear one, but day one you should definitely have this in your pocket if you're going into dental hygiene, please. Here's a nice picture of that. You can see the incisors towards the median portion of the mouth, the canines off to the side, those pointy ones, there's 4 usually, and then you get into the premolars and the molars as you go back. Here's the section of the tooth. Crown above the gingiva, above the gum line, the root sitting below, and then with the dentin, the pulp, and those blood vessels. We're going to get much more into this when we get into lab, so that's why I'm kind of going quickly through that. All right, so salivary glands. Still in the oral cavity, the salivary gland secretes saliva. Don't get your salivary glands mixed up with your tonsils. Okay, we already did tonsils. Those are lymphatic tissue for immune function. Salivary glands have this digestive function. So they're secreting saliva, which is a mixture of different things. It's used to moisten dissolved food particles, binds them together, helps in tasting. When things are moist, they have a stronger taste to them. It helps with cleaning the mouth and teeth. There's lysozyme in there and IgA in your saliva to help with any kind of pathogens. So salivary glands, the gland tissue has serous cells that are producing that watery fluid. It has salivary amylase, and then your mucus cells produce mucus. When you mix mucin with water, you get this mucus. And so it's this mixture of those two things, of mucus and this salivary amylase fluid. Amylase is going to break down your starches. So that's one of those things you'll need to learn, these enzymes. Anytime you see ACE, that means it's an enzyme. So amylase is breaking down starches. Proteases break down proteins. Those are a little bit easier. With some of these starch enzymes, it can be a little bit tricky. Salivary glands receive parasympathetic stimulation, so that rest and digest division of our nervous system helps to increase the production of saliva and water. And so when you are stressed, your body's going to conserve water, your mouth gets dry, right? That's part of that idea of being nervous, is your mouth kind of dries out. But when you are relaxed, right? When you're sleeping, you start drooling because you're receiving all these parasympathetic signals. Digestion is very active when we're resting, and so you get a lot of saliva production, even at the thought, sight, or smell of food. The first step in the digestive process is actually just thinking about your food, seeing your food, and smelling your food. That aspect of knowing that you're about to eat and what you're about to eat, your body goes ahead and starts to release different enzymes for that and preparing your body for it. So the major salivary glands, the 3 major salivary glands, the parotid, submandibular, and sublingual. So the parotid are the largest. They sit right in front of the ear, secrete that watery serous fluid, lots of amylase in that. Submandibular, they're on the floor of the mouth, so submandibular, think about that name and where it tells you where it is. They have more viscous fluid, so it's a little more serous and mucus than the parotids, where the parotid tends to be pretty watery. And then you have sublingual under the tongue. Smallest of all of those, thick and stringy, so it's mostly mucus from your sublingual glands. Here's a nice depiction of that. You have the submandibular under that jawbone, the sublingual under the tongue, and then the parotid just in front of the ear. And you can see this duct right here leading to it. So these things have ducts. So are they endocrine or exocrine glands? You tell me. All right, your pharynx. Your pharynx is your throat. We've talked about this a little bit in lab, I believe. So it is just posterior to the mouth. It technically starts in the nasopharynx, right? That upper portion behind the nasal cavity goes through the oral cavity and then down into the larynx. And so they're just named naso, oro, and laryngo, depending on where they might be. It is a tube. It's connecting those structures. The esophagus is going from the pharynx to the stomach. So when you swallow, things move into your pharynx, your oropharynx. It moves past the laryngopharynx, and then it enters into the esophagus. It's one continuous tube. Here you can see that the larynx anterior, the larynx and the trachea anterior, and the esophagus posterior, kind of flattened space right through here. So looking at swallowing, so as material is moving from the oral cavity into the pharynx, there are 3 stages to it. The first stage is a voluntary stage. That's when you're chewing food, you're mixing it all up, you're getting it ready, you're breaking things down. We call it a bolus, right? And then you start to move that towards the back of your mouth. You use your tongue to kind of push that towards the back of the oropharynx. In this second stage, the receptors in the oropharynx notice that food, and that starts to trigger the swallowing reflex in the medulla oblongata. When that happens, your soft palate and your uvula elevate to block the nasal cavity, and the hyoid bone and larynx elevate, and the epiglottis closes the larynx. So you have the uvula blocking the nasal cavity and the epiglottis blocking the larynx. We also use the tongue to press up against the soft palate, sealing off the oral cavity, so that way it kind of blocks things down, so there's only one exit for the food. Your throat muscles start to contract. The larynx starts to relax, opening up the esophagus a little bit more. And you have these waves, those peristaltic waves into the pharynx, and breathing simply stops. So usually, as we age, we get really good at being able to time breathing with swallowing. When we're younger, that's why kids tend to choke a lot, because they aren't very good at timing their breathing with their swallowing. It's truly it's a reflex in their nervous system that just isn't as well developed. And then the third stage, you have that transport food from esophagus to the stomach. So that continuing of moving down. So the second and third stages are involuntary. Once we get into the esophagus, it is a straight collapsible food passageway leading to the stomach. Collapsible. So that means that when there's no food in there, it is not opened up. It's about 25 centimeters long, sitting behind the trachea. It extends down through an opening in the diaphragm called the esophageal hiatus. So your stomach is in your abdominal cavity, not in your thoracic cavity. And so to get to the stomach, the esophagus has to pass through the diaphragm. And so an opening is called a hiatus. Sometimes you'll hear about people having a hernia here, because part of the stomach will move up through this opening, up through this hiatal opening. And they call it a hiatal hernia. There are mucus glands all through your esophagus to help keep things moist and keep things lubricated. And you have what's called the lower esophageal sphincter, or the cardiac sphincter. This is making sure that things don't go from the stomach back up into the esophagus. And when people have acid reflux, typically what's happening is that some acidic content from the stomach is pushing back through this sphincter. And we call that heartburn, one, because it's right by your heart. That's if you've never had heartburn before or acid reflux before, then it's kind of hard to say. It really is like this burning sensation that kind of sits right behind the heart. And for some people, it can get severe enough to where they think they are having a heart attack. So your stomach itself is a J-shaped organ in the upper left abdominal quadrant. It has these features called rugae. We call them gastric folds is the other name for them. And it's these ridges on the inside of the stomach, these features of the mucosa. And that allows the stomach to stretch and to draw back in. So whenever you see the inside of a stomach, it always has these ridges on the inside. It's because it's kind of like elastic that allows it to expand. The stomach is receiving food from the esophagus, mixes it with digestive juices. It starts to break down proteins. The acidic nature of the stomach can help with that. There is some absorption of nutrients, not a whole lot. Its main function is to have this mixture of digestive juices and kind of breaking food down even further to allow more exposure of surface area so that things like the HCl, the hydrochloric acid, and the stomach enzymes can start to break things down a little more. And then it moves it to the small intestine. The stomach has multiple regions. We'll look at these in lab. The cardia is close to the opening of the esophagus. The fundus is this kind of rounded portion just inside of the cardia. The main region is called the body. And then the lower portion, the distal portion, is called the pylorus. And this is what is going to be moving food to the small intestine. And so food passes through these four regions in this order. First it comes through the cardia, it moves over to the fundus, it goes down through the body, and then it moves out through the pyloric part. The pyloric sphincter, this is the other sphincter in the stomach. You have the cardiac sphincter, the lower esophageal sphincter, whichever one you want to call that, at the opening, and then the pyloric sphincter is at the exit. And so it controls movement of food from the stomach to the small intestine. So here's your picture of your stomach. You see you have the esophagus with the cardiac sphincter right here. You go again into the cardia, the fundus, the body, and then the pylorus is this kind of bulging portion down here. And you can see all these rugae in the middle. The pyloric sphincter leading into the duodenum, which is the first part of the small intestine. So looking at these gastric secretions that I was talking about, your stomach is lined with gastric glands within the mucosa that open into what's called gastric pits. And gastric pits kind of are lined with all these different cells. You have mucus cells making mucus, so that's for protection because the parietal cells are secreting hydrochloric acid. Hydrochloric acid is what keeps the stomach pH at around 2 or 3, and without this mucus layer, that would start to degrade and break down your stomach lining. And when people get ulcers, it's because this mucus layer is being degraded, and the HCl is getting to make direct contact with our body cells, and it starts to break them down and expose blood vessels. So the mucus cells are producing mucus for protection. Chief cells are secreting pepsin to digest proteins. First, it starts out as pepsinogen. So whenever you see something with ogen in it, it means that it's some type of enzyme or some type of inactive form of a chemical, and so it needs to be activated. So pepsinogen is activated into pepsin by the low pH of the hydrochloric acid. This is good because otherwise if protein enzymes were always available or always active, then they would start breaking down our body cells. And so only having them active in certain conditions is a way to protect ourselves. As I said, parietal cells are secreting hydrochloric acid to activate pepsinogen for destroying any bacteria. It makes a bacteriostatic environment, as well as intrinsic factor, this molecule called intrinsic factor, which actually is not active in the stomach, but is required for B12 absorption from the small intestine. So intrinsic factor just kind of hitches a ride all through the stomach, all through the small intestine, and then in the small intestine, it binds with B12 to absorb it into the stomach, or into the blood. Enteroendocrine cells, enteroendocrine, right? Let that name tell you what it is. An endocrine cell is a hormone-producing cell. Entero is another prefix for the digestive system. Specifically for your kind of intestinal systems. So when you see entero, you should be thinking GI tract. So enteroendocrine cells secreting hormones acting locally, things like gastrin to help increase that stomach action. Altogether, mucus cells, chief cells, parietal cells form your gastric juice. So when we talk about food being mixed with the gastric juices, it's the secretions of these cells. Here you can see the gastric pits lining the mucosal layer, and then all the different kinds of cells that are lining them along in here. Here also is just more breakdown, component, source, and function. What's being secreted, where it comes from, and then what is its role. Right, regulation of gastric secretions. So for the most part, again, parasympathetic activity is going to be increasing your GI tract. Sympathetic will be decreasing it. Gastric juice production occurs almost continuously, but can be modified neurally and hormonally. Secretions are going to be enhanced by parasympathetic impulses. Acetylcholine and the hormone gastrin both help to increase stomach activity. Once foods start to get into the small intestine, that increase of stomach acid, that increase of the acidic contents being released into the small intestine actually triggers sympathetic impulses to slow down the digestive process. This is just to make sure that it's not happening too fast. We'll see over and over again that as one part of the digestive tract gets activated, another part further down will be told to move it along, and then the part further up will be asked to slow down a little bit. This is to make sure that there is never too much overcrowding of the GI tract. That's why things can sit in your stomach for 2 hours up to 12 hours sometimes. Same thing in your intestines. As soon as you start to kind of think about eating, your GI tract will start to move things along through your intestinal tract to make more room for stuff to be continued to pass. Even things like the presence of fats and proteins causes a release of cholecystokinin. We'll call that CCK, cholecystokinin, and that decreases gastric motility as well. So again, this idea that as food gets moved into the small intestine, the body's responding to it, and it's saying, okay, just tell into the stomach, give us a moment, let us figure out what we got here, and then we'll let you know when it's time. Once these levels of CCK and the sympathetic impulses start to decrease, then you start to get more movement, more gastric motility. So there's just a nice little depiction of that. You start with the parasympathetic impulses, increasing motility, stimulating the release of gastric juices, release of gastrin into the bloodstream, and then gastrin increases stomach activity as well. And so this is definitely increasing, it's almost like a positive feedback here. So in the stomach, within the stomach, we are absorbing water, some salts, alcohol is absorbed here, and then some lipid-soluble drugs. Only small quantities, so not really a lot. When I say water, it's a very small amount of water that gets absorbed here. The large majority of water is going to be in the large intestine. All right, mixing and emptying actions. So after a meal, the mixing actions of the stomach turn food into a pasty mixture of food and gastric juice called chyme. So that chyme is the mixture of your ingested food mixed with all these gastric juices. Through peristaltic movement, that chyme is pushed towards the pyloric region. It kind of pushes up against the pyloric sphincter, and that will cause it to be stretched a little bit, so it actually goes into the duodenum in small amounts. It's not like your whole stomach empties at once. It just releases small little amounts at a time. It really does depend on the fluidity of the food. Liquids are going to pass through a little bit more quickly. Fats can be in the stomach for 3 to 6 hours, and proteins and carbs can pass through quickly as well. So depending on the mixture of food that you're eating, that will determine how quickly it's going to be passing through. As chyme fills the duodenum, those accessory organs, things like the pancreas, liver, and gallbladder, start to add secretions to the mixture. So it's really in the duodenum and the jejunum that we have a lot of the major actions of chemical digestion and absorption happening. Let's talk about the pancreas, though, because it is going to be necessary when we get into small intestine function. We've looked at the pancreas as an endocrine gland, so now we're looking at it as an exocrine gland. Remember, its endocrine functions are blood sugar regulation, insulin and glucagon. The exocrine function is to make pancreatic juice. So we had gastric juice, now we have pancreatic juice. So it's a mixture of different things. The pancreas, because it's so closely associated with the small intestine, like both literally, they are kind of just right next to each other. They're attached to each other in a way. As as chyme moves into the small intestine, to the first part, the duodenum, the pancreas is going to pick up on the contents, and it's going to release its juices for that. So these juices are made in the acinar cells of the pancreas. They release into these tiny tubes and eventually give rise to the pancreatic duct. The pancreatic and bile ducts join together, and they both form the hepatopancreatic sphincter. Hepatopancreatic, liver, pancreas, sphincter. This is what connects these two ducts into the duodenum. So here you can see that. There's your gallbladder, your bile duct, merging with the pancreatic duct into the hepatopancreatic sphincter. Right in there. So looking at pancreatic juice, it does have some different enzymes for digesting carbs, fats, proteins, and nucleic acids. So all your major macromolecules are going to be affected by your pancreatic juices. It's not like there's one thing though, right? We have different enzymes for each of those. So you have amylase and lipase. Amylase for carbs, lipase for lipids, lipase. Nucleases, breaking down nucleic acids into their nucleotide monomers. And proteolytic enzymes, proteolytic, protein, lytic, lysis, breaking apart. Trypsin, chymotrypsin, carboxypeptidase. You need these three different ones because they're each going to take care of different bonds in the amino acids. Proteins are large, complex structures. They have a lot of different bonds in them. And so you need all three of these to be able to isolate the different bonds that have to be broken apart to actually make an amino acid monomer. And that's required for absorption. You can't, again, absorb an entire protein. You have to absorb it in its monomer form. And that goes true for any of our macromolecules, nucleotides, triglycerides, and starches. They all have to be broken down into their monomer forms. Just like in the stomach, the proteolytic enzymes in the pancreatic juice are stored in an inactive form in these vesicles in the pancreas to make sure that they're not actually digesting the organs in the pancreatic cells. They get activated once they're in the small intestine. The last thing, the fifth thing in pancreatic juice is bicarbonate ions. They create a buffer for the hydrochloric acid. So the stomach acid should be around 2 to 3 in terms of pH. That's a lot of HCl. A lot of other cells can't survive that kind of pH in our body. And so once chyme gets moved into the duodenum, your pancreas is going to start releasing bicarbonate to buffer that. Right, so regulation of the pancreatic secretion then. You have, again, nervous and endocrine systems helping to control the release of pancreatic juices. Parasympathetic impulses increase the release. You also have, when acidic chyme enters the duodenum, secretin from the duodenum stimulates the release of pancreatic juice with bicarbonate ions inside of it, but not a whole lot of digestive enzymes. So if you are maybe just drinking a liquid, let's say you're drinking Coca-Cola, that's already very acidic, and then your stomach is also very acidic, and liquids move quickly through the pancreas or through the stomach. That's pretty acidic liquid that's being moved into the duodenum with very little things other than some amylase to be broken down, some sugars to be broken down, which you already have salivary amylase doing that. So for the most part, this is when you would have a lot of bicarbonate ions being secreted from that secretin hormone stimulation. When it comes to fat, proteins and fats, then it's cholecystokinin. You have CCK from the small intestine gets released and stimulates the pancreatic juice with abundant digestive enzymes. So now let's think maybe we're eating a pizza. You have a nice, fatty, greasy slice of pizza that's got that thick carb crust, right? So then you're going to need secretin for the acidity from all those tomatoes and things from your stomach juices, and cholecystokinin to help break down the proteins and fats. There's a nice little depiction of that. You can kind of walk through the process of chyme coming in and then how secretin works. All right, and then let's look at the liver and the gallbladder. So again, this is when we talked about the bile duct coming from the gallbladder. Bile is produced by the liver, so we're going to look at how they function in this process. So the reddish-brown liver, it's in the upper right quadrant of the abdominal cavity. It is the largest internal organ in the body. It has large right and left lobes, and then also these smaller quadrate and caudate lobes. So 4 lobes total, right and left, quadrate and caudate. These are anterior posterior lobes. And then once we get into the liver, it's separated even more. Each of the lobes is separated into lobules, that have these cells coming out from a central vein. So each one is supplied, has its own little individual blood supply, and goes through the functions of filtrate. We'll see the functions of the liver. There's a lot of functions in the liver. And you have these sinusoids. These are channels filled with blood separating groups of hepatic cells. We'll look at a picture of all that in a minute. Here you can just see the large liver. You have this larger right lobe, smaller left lobe, and then you have your quadrate. It kind of looks like a 1, 2, 3, 4, right? It has 4 sides, so that's a quadrate. And then this caudate lobe back here. So the caudate lobe is posterior. It's right beside the vena cava. The quadrate lobe is right beside the gallbladder. That's how I usually remember those two. So gallbladder, quadrate, caudate, vena cava. C for cava, if that helps. So looking at the liver structure and how it functions, the liver is responsible for receiving blood, filtering through it, removing triglycerides as it needs to, synthesizing cholesterol. It has a lot of different functions. One of those big functions too is producing bile. So let's look at the vessels that the liver has to be able to do this. You have an artery. So the hepatic artery is oxygen-rich blood to the hepatocytes. This is what is giving them the nutrients that they need to do their jobs. You have the portal vein. The portal vein is coming from your intestines. It has oxygen-poor blood, but it's rich in nutrients to the liver for adjustments to nutrient concentration. So the liver is really responsible for making sure that nutrients are distributed evenly through the body. So hepatic artery, this is coming from your cardiovascular system, oxygen-rich blood to the hepatocytes. Hepatic portal vein is coming from the intestines, oxygen-poor, nutrient-rich. Then you have the bile duct. The bile duct is transporting bile from the hepatocytes to the bile duct. So the artery and the portal vein are both going in towards the hepatocytes. The bile duct is moving away from them. Blood passes from the sinusoids into the central vein of a lobule. A sinusoid, by the way, is a leaky capillary. This is referring to blood vessels that have a lot of kind of leaks and holes in them, so that larger vessels can start to kind of get out of there. Blood passes, again, from the sinusoids to the central vein and exits through the hepatic veins that eventually merges into the vena cava. You have Kupffer cells. These are a type of macrophage that are performing phagocytosis in the liver. If you remember when we talked about macrophages, depending on if once they enter a certain organ, they get called something different. In your skin, they're called mast cells. In the liver, they're called Kupffer cells. So the bile secretions from hepatic cells collect into bile canaliculi. Those form bile ductules, hepatic ducts, eventually the common hepatic duct, and then that transports it out of the liver. So easier to look at a picture of this sometimes. So here you can see the different lobules of the liver, these different kind of hexagonal structures. Each one has a central vein, and then you have these other sinusoids kind of all around it. And then out here you can see the triad. So the triad is both the hepatic portal vein, hepatic artery, and the bile duct. So blood, and here you go. Here we can look at this section of it now. So you have your hepatic portal vein and your hepatic artery, both sending blood to the central vein, and then your bile duct. As blood comes through here, it gets filtered, right? This hepatic artery is giving blood to the hepatocytes. Things are being filtered out. The hepatocytes then are making bile, and that's what all this is. This is all bile. So bile moves, I think this next slide, yeah. Bile moves away from the central vein, while all of these are moving towards the central vein. And then again, this eventually goes out of the liver to the vena cava. All right, so these functions of the liver. So liver is a crucial organ in our body. It's actually one of the only organs in our body that can regenerate itself. When you go to have a liver transplant, they will take part of your liver and put it into somebody else. That part will grow into a liver, and then yours will regrow itself. It's pretty wild. And the liver can pretty much function fully up until about I think you can get down to 20%, 10% of your liver function before your liver actually starts failing. So your liver has a lot of redundancy built into it to make sure that it can continue these functions. So it's responsible for a lot of metabolic activities. When we say metabolism, right, we're talking about breakdown typically. The metabolism of carbs, lipids, and proteins. Proper blood concentrations of blood glucose and nutrients. It helps to do that by storing glycogen or releasing bound glycogen. It stores vitamin A, D, B12, iron. People eat liver and onions to get their iron back up. It synthesizes lipoproteins, phospholipids, and cholesterol. Cholesterol is super important for our cells. Phospholipids, those can be on cell membranes or other structures we find them for activating things. Converts excess carbs and proteins to fat for storage. Deanimates amino acids to make urea. Synthesizes plasma proteins and clotting factors, so things like your albumins and globulins for transporting different substances, hormones, things like that, antibodies. Converts amino acids to other amino acids if they are needed. It filters the blood. It removes damaged blood cells, foreign substances, removes toxins. It has enzymes for detoxifying different things. So your liver detoxifies. Let's talk about that. You do not detoxify by sweating things out. Your skin does not detoxify things. Your liver has certain enzymes that aid in detoxification. So that's, sorry if I got a little intense on that, but not everything is just going to be detox. You can't do a juice cleanse and it be a detox. That is not a detox. Not how it works. Role in digestion is to secrete bile. So all of these other things that it's doing, yes, this is all helpful, but when it actually comes to how does it help with digestion, it's by secreting bile, by producing and secreting bile. So this is a good table to be familiar with. General function, and then what does it mean specifically? So yes, it does storage, but specifically it stores these different things. Let's look at bile now. So as we said, bile is the main product of the liver for digestion. It's a yellowish green liquid that the cells secrete continuously. Just saying, when you throw something up when you're vomiting, a lot of times people say, oh, I threw up bile, I have this bile taste. You're not throwing up bile. Bile is being released into your small intestine. When you throw up, what you're throwing up is gastric juices. That's why it has such an acidic taste to it. Bile, I mean, I've never tasted bile before, but I don't know why it would be acidic. It doesn't have anything in there that really makes it acidic, and our body doesn't need to have anything that acidic. So when you're throwing something up and you get that taste in your mouth, a lot of people say, oh, it's that bile taste. And they say that because they know bile is like a yellowish green, and the stuff you throw up tends to have a yellowish color to it. It's not bile. It's gastric juice. It's full of HCl, hydrochloric acid. That's what makes it acidic and so bitter. So your bile is composed of water, bile salts, pigments, cholesterol, and electrolytes. The pigments, bilirubin and biliverdin, are from hemoglobin breakdown and from old worn-out blood cells. If you remember, we talked about how blood cells are recycled, and so these are two pigments that come from the breakdown of hemoglobin. Out of all of this, it's just the bile salts that have digestive function. So bile, while it is aiding in digestion, really like the bile pigments, the things that are here, this is going to be secreted as waste. This goes out of the body. Same thing with cholesterol, excess electrolytes, or these can either get absorbed or excreted depending on what the body needs. So it's not like all of this is for digestion. It's these bile salts that are functioning in digestion. We'll see how they work. You may have heard of jaundice. Jaundice is a yellowing of the skin and the mucous membranes, and this is because you have an accumulation of bile pigments. So the bilirubin, biliverdin, rather than being mixed in with bile and secreted out, they get stuck in the liver. They get transported back through the blood, and it causes that this yellowing color. This could be a blocked bile duct or gallstones blocking the gallbladder, tumors, cirrhosis. This is liver failure. Hepatitis, rapid red blood cell breakdown, so if your body is just for some reason going through a lot of your red blood cells, or transfusion of incompatible blood. So the gallbladder. We did the liver. Liver produces bile. The gallbladder stores bile. So yes, your liver can produce bile and then you just immediately secrete it, and that's what happens to people that have had their gallbladders removed. If you have had a cholecystectomy, your cholecyst is your gallbladder. If you've had your gallbladder removed, your body doesn't store bile. It is produced by the liver, and then it is immediately secreted into the small intestine. So people that have had their gallbladder removed really have to regulate their fat intake. because bile salts are mostly for breaking down fats. That is their primary function. So the gallbladder is this little sac just underneath the liver on the inferior surface. It stores bile between meals and reabsorbs water to concentrate that bile. It contracts as a muscular wall to contract to release bile when it gets signaled through the cystic duct, forming. It's connected to the cystic duct. That's the one that comes directly off of the gallbladder. The common hepatic duct, that comes from the liver. Those two merge. These two ducts merge to make the bile duct, or the common bile duct, if you want to call it that. This is the one that actually leads to the duodenum, joins the pancreatic duct with the hepatopancreatic sphincter, and gets released into the duodenum. Certain conditions, cholesterol can precipitate, meaning that it hardens and it kind of clusters together, like salt precipitates out of water, right? When you dehydrate something and you're left with just the salt on top of it, that's what precipitating means. So cholesterol can start to actually clump together and form gallstones that then have to be removed. So how bile is released, that regulation, it is typically just under the stimulation of CCK. So as CCK, cholecystokinin, is released in the presence of fatty foods, then that stimulates, it goes to the bloodstream and it stimulates the gallbladder and liver, I think, to release bile. The hepatopancreatic sphincter remains contracted unless a peristaltic wave approaches, and then it starts to relax and bile enters the duodenum. So once that wave starts to come in, that's when it starts to soften and release. Otherwise, it would just be releasing bile constantly, and we would be wasting it. Here's a depiction if you want to walk through these steps to see how bile is produced or is released. There's a radiograph with gallstones. They would not actually appear that green color. I don't believe. I'm pretty sure not, but I'm not a doctor. As much as I do know, I'm not a doctor. So somebody can check me on that if you want to. All right, hormones of the digestive tract. So main ones: gastrin, cholecystokinin, secretin, source and functions. All right, so now let's look at bile salts. So we said bile salts are the only ones that really function in digestion. They themselves are not enzymes. What bile salts are going to do is work in a mechanical function to accomplish what we call emulsification. This is a mechanical process, not a chemical one. If you remember me saying that for digestion to really be optimized, you have to break down things into smaller portions to increase the visible surface area. If you think about one big piece of pizza, right, that's, it only has so many approach points. You're not just going to lean down and, you're not going to be a monster and just bite a chunk out of the middle of the pizza. But if you take that pizza and you cut it into 8 different slices, well, now you have the outside of the crust, you have all along the sides to the tip, you have all these different points of access. And so the more you break something down, the more surface area gets exposed, and the easier it is for enzymes to digest stuff. So bile salts help do that by taking lipids and putting them into smaller and smaller little groups so that the lipases can then work more effectively. This is called emulsification. So emulsification, you're breaking down large fat globules into smaller droplets that increases their solubility. Increases the surface area of fat so that droplets can disperse in the water, makes chemical digestion by enzymes called lipases more effective. And then the bile salts aid in absorption of fatty acids, cholesterol, and fat-soluble vitamins. So pretty general from what I just said. Sorry, I thought we were going to go a little bit more into fat absorption there for a second, but we are not. So I want to make sure I didn't want to say anything else about that. And if you think about like when you're washing dishes, right, isn't that one of the things like Dawn Dish Soap always talks about? Oh, breaking down, sticky substances, fatty substances, all that kind of stuff. What you do as a person washing dishes is that you scrub first. You probably rinse some water over it. You might run a little towel over it or washcloth sponge, whatever you use. And then you might add in some soap. You might add in that there. And it's like, oh, wow, look how quick this unsticks just with some soap. It's because it's a physical process that then allows this chemical action to occur. And so emulsification by coating fats in these salts it allows them to be swept away through water easier. And so that's what liquid detergents do, is that like the Dawn dish soap, at a microscopic level, it's actually getting in between food and the plate, and it's kind of slipping in there and then making this nice little barrier that can then increase the solubility. You can just wash it off, wash it away. That's why it's good to wash your hands with soap and water. Don't just rely on hand sanitizer. Hand sanitizer is alcohol based, that's water soluble. It does not actually break down the membranes of certain bacteria. With those, you need to have a emulsifier. You need to have a soap based detergent to be able to get them to wash off your hands. Otherwise, you can sit there and just run water over your hand and some bacteria will stay. All right, back to digestion. Sorry about that. So, small intestines. So we've moved from the stomach. We had all our accessory organs releasing their juices. Now we're moving food through the small intestine. Small intestine is a long tubular organ, going from the stomach to the large intestine. Fills a large part of the abdominal cavity. It is longer than your large intestine, but it's smaller in diameter. So the main functions is receiving chyme from the stomach, pancreatic juices from the bile and from the pancreas, bile from the liver and gallbladder. It finishes digestion of nutrients that arrive in the chyme. So again, chyme is partially digested in the duodenum, in the small intestine rather, not just the duodenum, but in the small intestine. This is when that process gets completed. They get broken down into their small monomer forms and then can be absorbed. And so most absorption happens in the small intestine, and then anything remaining gets transported to the large. Three main sections of the small intestine: the duodenum, jejunum, and ileum. Duodenum is the shortest. It's what connects to the stomach. The jejunum is the largest, makes up 2/5 of the small intestine, slightly larger diameter than the ileum, more active in the digestion process. So most digestion occurs within the jejunum. The ileum is the more distal portion connecting into the large intestine. The jejunum and ileum have what's called a mesentery. This is just a covering. It's a double layer of the peritoneal wall of the visceral peritoneum that essentially just adds extra protection. It has blood vessels, nerves, lymphatic vessels, and there's also something called the greater omentum draping over the intestines. So here you can see the mesentery. This is within kind of that jejunum ileum area. There's the duodenum right at the top. And so the mesentery is there with all its blood vessels holding things kind of in place. And then what you're not seeing is the greater omentum. The greater omentum is kind of like an apron that I'm kind of running my mouth side to side to kind of show that it would kind of go side to side, and then it hangs down and it just kind of covers everything and acts as a wrapping. Here is a side view of the digestive tract. This can be a little confusing at first, so it's nice to sometimes have it labeled and to go through all of it. You can see liver, you've got your pancreas back here with the stomach. You get into your intestines, so these are all the different folds and things of the small and large intestines. Then you get into, going backwards, you get into the rectum and the anus back here. I started to go down to the bladder and I was like, wait a second, we're going out the wrong end. We want to be going out this way. All right, structure of the small intestinal wall. So we talked at the beginning of this lecture about some modifications that we'll see on the intestinal wall, and those really are present in the small intestine. So the small intestine has those villi, those intestinal villi, to help increase surface area for absorption and mixing. Each villus, as we said, has that simple columnar cell that's going to be covered in microvilli. And inside of that large villus, you're going to have a lacteal, which is from the lymphatic system for transporting fats. Blood capillaries and the lacteals transport absorbed nutrients out of the alimentary canal and into the body. So once things enter the blood vessels, the capillaries, the lacteals, that's when they're technically inside the body. You have intestinal glands as well that are going to be releasing certain juices, certain secretions, and these cells are typically replaced every 3 to 6 days. So here's just a basic example of that. Here are these intestinal glands down in here doing things like mucus. You have globate cells for mucus as well. You have this large lacteal, this green lacteal moving up the center of the villus, surrounded by these capillaries, arteries and veins for absorption. So these arteries, these blood capillaries are taking fresh blood to the villus, and then the veins are going to be absorbing any nutrients that are water soluble, and then transporting it back to the hepatic portal vein, where it goes to the liver for filtration, and then into the heart for circulation. There's just a micrograph of that. All right, secretions from the small intestine. Mucus, as we said, coming from the goblet cells, all abundant through the mucosa, keeps things moving, keeps things sliding along. You have this watery fluid from the intestinal glands, again, just carrying products into the villi, transporting things out of the body, out of the digestive tract. You have different enzymes, so just the ones from the pancreas weren't it. You do have peptidases breaking down dipeptides to amino acids, sucrase, maltase, lactase for different saccharides, and intestinal lipase for triglycerides to fatty acids and glycerol. So again, everything getting broken down to its monomer components. Here are a nice little chart. Here is a nice little chart of all of that. That salivary amylase, just because it was produced in the mouth, it gets deactivated in the stomach, but it can be reactivated again. You have pepsin for proteins, lipases for fats, and then these proteolytic enzymes, again, all three of them for breaking down those different bonds in the protein chain. And then a few more from intestinal mucosal cells. You can see those a little bit more. Peptidases, proteins. These are all of your amylases, different types of amylases. Intestinal lipase, and enterokinase. This is converting that trypsinogen, that one of those peptidases, into its active form. All right, so absorption in the small intestine. So as we said, digestion is completed in the small intestine, and then absorption can really start to occur. So major site for absorption is the small intestine through those villi and microvilli. Very few absorbable nutrients reach the distal end of the small intestine. So that's just meaning that by the time you get to your ileum, most of the absorbable nutrients, the nutrients that can be absorbed, have been absorbed. Monosaccharides are absorbed through those carbohydrate monomers. Those are absorbed to the villi through active transport or facilitated diffusion. So this is an energy-consuming process. They go into blood capillaries. They are water soluble. Amino acids absorbed into blood capillaries by active transport as well. These are also water soluble, so they're transported through the blood. Fatty acids and glycerol, so the monomers of your triglycerides, from your lipids, right? So lipids, one form of lipid is a triglyceride. A triglyceride is of 3 fatty acid tails and a glycerol backbone. Sorry, pollen's getting to my throat. Those all diffuse into the villi, and these are actually going to be transported through the lacteals. So fatty acid absorption. The endoplasmic reticula of the cell, so endoplasmic reticula, reticulum, those packaging centers of the cells, use fatty acids to resynthesize triglycerides. So what happens is that your body, in your small intestine, it's going to break down triglycerides into the monomers, fatty acids and glycerol. Your cell will absorb fatty acids and glycerol, and then it actually puts it back together into a triglyceride. So your cell can't absorb the triglyceride as a whole, but it can export it. So your body breaks it down, puts it back together inside of the cell, resynthesizes the triglyceride form. Those start to get encased in proteins called chylomicrons. And then the chylomicrons are absorbed by lacteals traveling through the lymphatic system. So it's a step-by-step process. Again, monosaccharides and amino acids are both water-soluble, so it's easy. But because triglycerides are not, they break it down into its monomer form, absorb it into the cell through those villi. It gets reconstituted as a triglyceride, packaged into chylomicrons, these protein kind of capsules, and then it's absorbed by lacteals. All right, and then let's follow those lacteals. Let's follow those chylomicrons. So they get transported in lacteals through the cardiovascular system, and eventually they make their way back to the liver. What are they doing though? So chylomicrons transport dietary fats to muscle and adipose tissue. You have three different kinds: VLDL, LDLs, and HDLs. If you've ever gone and gotten blood work, or you have someone in your life that is on cholesterol meds, you probably have heard of some of these terms before. So VLDL, very low density lipoprotein. These are produced by the liver, transporting triglycerides from the excess dietary carbohydrates. At the adipose cells, those start to unload the triglycerides. So basically, this is acting as storage. Let's say that you are having excess intake of these nutrients from your diet, and so your body has to do something with them. So it's going to take them and it's going to put them in adipose cells. Once it does that, once it kind of unloads those triglycerides, it starts to then leave LDLs. So VLDLs, those have triglycerides. Those are the ones that hop off first. And then you have LDLs that circulate through. LDLs are mostly cholesterol. So LDLs are considered quote unquote bad cholesterol because this is where it gets confusing because we start to talk clinical and textual. So it's going to call all of this cholesterol. When you go to the doctor, they talk about your good cholesterol as your HDL and your bad cholesterol as your LDL. But really, these are lipoproteins. They're not cholesterol molecules. So just kind of keep that in mind. LDLs, though, the higher the number of LDL that you have in your blood work, it's considered more risk. You're considered at higher risk because LDLs are mostly cholesterol. And they're taking it and dropping it off at cells. HDLs are high density. So once the LDLs have dropped off their cholesterol, they're now coming back to the cells. They're coming back to the liver. And so the more HDL that you have, it's considered good cholesterol because these are actually removing cholesterol stored in your cells back to the liver for being processed out of the body. So VLDLs transporting triglycerides. They then become LDLs, which are transporting cholesterol. And then you have HDLs, which are removing cholesterol back to the liver. So HDLs are the only ones that actually go from the liver or from the body to the liver. VLDL and LDL go liver to body. All right. There's also, so jumping off of fat transport for a little bit, villi also absorb water and electrolytes. Here's triglyceride reabsorption. So again, you can see fatty acids and glycerols are absorbed into the cell, repackaged as triglycerides, covered with chylomicrons, and absorbed through the lacteals. There's a nice little chart for you to help understand what is being broken down, how it gets absorbed, and where it goes. All right, so movement in the small intestine. So this is where a lot of the movement happens. You have that segmentation, that back and forth, breaking up chyme, mixing it with all those juices that we were talked about being released. And then peristalsis, these waves that help to move things through nice short distances. I'm just kind of pushing it along, shuffling it through the intestines. It can take 3 to 10 hours to travel the length of the small intestine. A strong peristaltic rush from overdistension or irritation. If that happens too quickly, it can cause diarrhea. This occurs because if things don't move slowly, if they're not given this 3 to 10 hours that they need, you don't have proper nutrient absorption, you don't have proper water absorption, and so it comes out really liquidy. And so due to all that lack of absorption, it causes this really quick movement, liquid bowel. What you have is the ileocecal sphincter at the junction of the ileum and the cecum, which is the first part of the large intestine. That stays closed unless you have a reflex after a meal. So as I said earlier, when you're eating, when you're digesting, as soon as you start thinking about food, as you start chewing food, swallowing food, that then triggers other reflexes further down the GI tract to help make sure that there is room for this new digested material we're about to try to process. So this ileocecal sphincter between the small and large intestines stays closed until you start to eat a meal. And then once you start to eat a meal, the gastroileal, gastro stomach, ileal ileum, this reflex is elicited, is activated, and basically it says it's the stomach saying, "Hey, ileum, I've got some food in here, so you might want to open up and push some of this stuff into the large intestine so we got some space." And that's exactly what happens. This opens up and it moves contents into the large intestine, that first portion being the cecum. And then we're in the large intestine. So the large intestine gets its name because of its larger diameter, even though it is smaller than the small intestine. Starts in the lower right portion of the abdominal cavity at the cecum. It ascends on the right, so it starts at the cecum, it goes up, it goes over, and then it goes down. Opens to the outside of the body as the anus, and the main function is to absorb water and electrolytes, form and store feces. There are a couple other portions to it that we'll see. All right, so first parts of the large intestine, the cecum is that pouch at the very beginning. And the cecum has this little tail called the appendix. You've probably heard about this. It is, for the most part, non-functional in modern humans. It does have some kind of immune function to it. There are little pyre patches in the appendix, and it seems that people that have their appendix removed might have some immune-compromised states to them. But for the most part, the appendix is non-functional, and most of us don't worry about it. Then you move into the colon. So a lot of times people will use colon and large intestine interchangeably, and that really is not correct. The large intestine consists of all of these regions. The colon only is the ascending, transverse, descending, and sigmoid regions. The colon does not include the cecum, the rectum, or the anal canal. So when we talk about the large intestine, we're talking about all of these bolded structures. The colon is just these 4 spots. So starting down here, there's your ileum. So there's the end of the small intestine. The ileocecal sphincter is here. So this is where food would be moved from the ileum into the large intestine. And technically at this point, we still call it a bolus. So this is where the bolus would be moved into the large intestine. The cecum is down here with that appendix. And then it's supposed to move up. It should go up the ascending, across the transverse, down the descending. And then you get into this little hook right here, this little kind of flexure, and this is called your sigmoid colon. So after you get through the descending, you start to move into the sigmoid colon, and this is where feces tends to sit. Fecal matter will tend to sit in the sigmoid colon until there's some other signals for it to move through. Once it gets from the sigmoid, it then moves into the rectum, and once it gets into the rectum, that's when you get that signal of, oh, I need to go to the bathroom. When it's in the sigmoid, you may not feel it. You may not get a signal. It could be one of those things, well, I could go try. Maybe I'll use the restroom. And it's like, oh, wow, I did need to go. Once it gets into your rectum, you know you need to go. You are getting signals. You are getting sensations. And you are consciously, we learn as adults that nerve reflex starts to form. And we learn how to control those voluntary sphincters to hold the fecal matter in. But up until that point, it's just kind of resting and hanging out in the sigmoid colon. Because of that, this tends to be the most common case for colon cancer, is in the sigmoid colon where fecal matter tends to just rest and stay. The rectum and then the anus are the final portions of the large intestine. You can see these kind of folds right here. This helps to slow down the movement of fecal matter as well as give us signals, those stretching signals that there is a need for defecation. In the anus, there are both internal and external anal sphincters. The internal anal sphincters you can see are a little more superficial. The external are a little deeper. The internal ones are involuntary. The external are voluntary. So the internal anal sphincters, you can see how far up they extend. That's why they're the internal. Their reflexes rather will extend up. And so they will sense this stretch and they will automatically release. And so at that point, it's just the external. It's that voluntary sphincter that we control that is remaining closed and holding material in. So you choose to relax the external anal sphincters to allow defecation to occur. All right, looking at the large intestinal wall, it does not have villi. The large intestinal wall is a little bit smoother. Same 4 layers, but has some unique characteristics. Not so much on the inside as much on the outside. Longitudinal muscles not uniform. They're arranged in this thing called the tinea coli, which extends the whole length of the colon. And the tinea coli is like a tendon, and as it contracts, right, or I guess it's muscle, so it's a muscle and tendon. Those things go together. So as it contracts, it creates these pouches called haustra, and this kind of creates little sections within the large intestine that fecal matter is arranged into. So I'm going to go back for a slide real quick, 2 slides. So you can see this thin line traveling up the outside. This is the tinea coli. And then each one of these little pouches, these little bulges, that is the haustra. All right, so functions in the large intestine. Does not absorb, digest or absorb nutrients. No real digestion or absorption of nutrients occurring here. Mucus is the only important secretion. Helps the remaining chyme pass along the intestine smoothly. Sometimes when we're dehydrated, you can't produce as much mucus, and so you have harder to pass bowel movements. What it does absorb are electrolytes and water. That is the main function of the large intestine, is to absorb electrolytes and water, one of the main functions. It has important bacteria, what we call our intestinal flora. So this is your native bacterial environment. This is unique to most people, unique to each person, and it is what helps to maintain your normal body health. A lot of people contribute this to also your mental function, your mental state. When your bacterial flora, your native flora starts to get thrown off, your body can start reacting in strange ways. You have a lot of nerves in your intestinal system, and so things can start to get fired in different ways if you don't have this native flora. They're responsible for synthesizing vitamin K, B12, thiamine, and riboflavin, all absorbed and used by the body. So your native flora is very important for that. Humans, we can't break down cellulose, so it passes as fiber, but normal colon bacteria can break down some of that. So the normal colon bacteria will actually break down the cellulose that we can't digest, and sometimes that is what produces methane. It releases that odorous gas. That's just looking at a micrograph of the large intestine mucosa. All right, so movements in the large intestine. So again, that's been a consistent throughout this lecture, is the movements of these different parts. There are some mixing and peristaltic movements of the small, the small intestine has, but they're a little bit slower. Mass movements can occur 2 to 3 times a day after a meal. That gastrocolic reflex. So kind of this shifting of big, again, if material is content, or content is being sitting in the large intestine, this is kind of a big shift to really make a lot of space for new stuff to come in. Defecation, stimulated by the defecation reflex, forcing feces into the rectum where it can then be expelled. Defecation reflex is done by what we call the Valsalva maneuver. It's holding a deep breath, contracting the muscles of the abdominal wall. So essentially, when you go to defecate, you kind of brace down. It's the same thing that we do that's used during labor and delivery. And truly what happens is that the epiglottis covers the top of the trachea, your diaphragm will harden, and it actually creates this really intense abdominal pressure to push things down and out. Which is why it's also dangerous to strain too much to use the bathroom. If you are not able to kind of sit and just, with mild effort, allow yourself to defecate, you don't want to push too hard, because that's how you can give yourself a hernia. You can strain something and you can get hemorrhoids. You can actually have ruptures of your intestinal wall because the pressure gets so intense. And then this is going to be our last slide. We're going to end on some feces. So feces is composed of undigested material, water. So still about 75% of feces is water, even if it does come out, semi-solid. Electrolytes, mucus, shed intestinal cells, and bacteria. The color of feces mostly due to bile pigments that were chemically altered by that bacteria as it was digested. And odor is due to chemicals produced by bacteria as they go through their functions. All right, thank you so much.